Hi everyone. This is part 2 of Bacterial Genomes. Part 1 was last week and I reviewed replication, transcription, and translation. I hope you were able to view it, and I hope you were able to view some of the videos that were uploaded into Blackboard that showed you the processes of replication and protein synthesis. All right, today I'm going to talk about bacterial genomes and what is a genome. We know it's a DNA, but I'm going to be talking specifically about the chromosomes of bacteria, their arrangement, their makeup, and plasmids. Before I go on specifically about bacteria, I want to compare the genomes of prokaryotes and eukaryotic cells and some of the similarities, but specifically the differences between both. All right, we know that prokaryotic cells have DNA, but many of the chromosomes are circular. In eukaryotic cells, they usually have more than one chromosome, and they are linear. So even the shape is different between the two. Next, chromosomes are in prokaryotic genomes are supercoiled. They're wound tightly and looped around proteins, not histones, but proteins. And they are packed in that area of the bacterial cell known as nucleoid. Eukaryotic genomes are wrapped around histones. So these are specific proteins in eukaryotic cells upon which the chromosomes are wrapped. And then they are packed into chromatin within, and that chromatin is within a nucleus. As far as transcription and translation, since there is no nucleus in prokaryotic cells, that process will take place simultaneously in the cytoplasm of the bacterial cell. Eukaryotic cells, on the other hand, will have transcription, which only occurs in the nucleus. And once transcription has occurred and you've got the messenger RNA, then that will be transmitted through pores in the nucleus out into the cytoplasm, where translation will take place. Next, bacterial genomes have little non-coding DNA. What is non-coding DNA? Well, that is DNA that basically are just nonsense sequence that are not expressed as genes. They're just basically a filler or junk. That's what they know right now. In eukaryotic genomes, the eukaryotic genome will contain tons of this nonsense or non-coding DNA junk. And some of the examples are seen in introns and repetitive sequences of DNA that do not express or cannot be expressed into genes. Finally, we know prokaryotic genomes may contain plasmids. In eukaryotic genomes, plasmids are not common. So just some differences between the two. Now, in prokaryotic cells, we do have different genome sizes, different amounts of DNA. And they measured in units called MB, million bases. Okay, that MB just gives you the length of the DNA. And if you look at the table here, we've got, it differs depending upon the type of bacteria. So the second bacteria down on the table, Bacillus subtilis, if you look at that, it has one circular chromosome. And in the parentheses, it gives you the length, 4.2 million bases. The Bacillus subtilis does not have any plasmids, though. So plasmids are not the rule, okay? They are contained in some bacteria, but not all bacteria. Many of the chromosomes are circular here. If we go down to Borrelia, that one, the chromosome is linear. Okay, and if you even move over to the plasmids, it has many circular and linear plasmids. So again, this just depends on the type of bacteria as far as the size of their chromosome and whether or not they have plasmids. Next slide. So how are these genomes organized? Well, sounding like a broken record, we know that the DNA or the chromosome, the DNA on that chromosome is arranged in a nucleoid, which is the area within the bacterial cell. Some additional information that DNA or chromosome is in the form of loops, and they all come from one area and origin of replication. So on the right hand bottom corner, they have the diagram of the bacterial cell. You're going to see the origin of replication. That's where the chromosome originates. That also is where replication, the process of replication, originates. And from there, you're going to see extensions in a loop-like formation. They are anchored together by proteins. That's what those green circles are. And one loop of that chromosome is called a domain. Another thing about the DNA as far as its arrangement is that it is supercoiled. Really wound tightly, supercoiled. So a little more about the nucleoids, the prokaryotic nucleoids, the DNA within that nucleoid. I did say that the DNA binds to architectural proteins. They are not histones. Histones are found only in eukaryotic cells. So these architectural proteins help keep the DNA organized into that looped formation. We do have enzymes, toposomerases, and they give you an example such as DNA gyrase, which introduces twisting and supercoiling, and also untwisting, uncoiling. Now, why would we need the DNA uncoiled? Well, for replication, to give an area for those enzymes to begin replication, or for transcription. So again, it is important that these enzymes keep it tightly wound, but at specific times to also unwind that chromosome. And this is just showing, A and B is showing what happens during to the chromosomes during certain stages of a population of bacteria. So in the exponential phase, when they are replicating like crazy, you will need some unwinding for replication to take place, for these enzymes to take hold and begin DNA replication. What happens later on at the stationary phase, where the bacterial cells are not replicating anymore, is that these enzymes are not being reproduced anymore. So the DNA is pretty much just, the chromosome is pretty much unraveled and unfunctional. So we count on these enzymes too to hold the form, the supercoiled form of the prokaryotic chromosome. So remember, I said that there's coding and non-coding DNA in prokaryotes. This is actually comparing a section of a prokaryotic genome to a section of a eukaryotic genome, the human genome. So the green bar at the top is a section of E. coli, the genome of E. coli. And the bottom, the purple bar, is a section of the human genome. Now what you pay attention to is the colors here. The green color represents gene. So this will be expressed as genes. Yellow is introns. These are non-coding or nonsense DNA sequence. And the purple is basically repeat sections, DNA sequences that are repeated several times throughout the genome, but they cannot be expressed as genes. They are not genes. So if you compare the top with the bottom, you're going to see that the E. coli genome is loaded, packed with genes. It doesn't have a lot of the purple or yellow, a lot of that non-essential or non-expressive filler. Whereas if you look at the human genome, well, we're filled with a lot of junk, okay? You'll see there maybe 2 or 3, small sections of green. We don't have a lot of genes compacted within our genome like bacteria. So bacteria cells are very efficient. Another shout out for those little guys. They may have a chromosome, but it is an efficient little chromosome. So I'm going to talk about 2 types of gene transfer in bacteria. We're very familiar with the first one, vertical gene transmission. In this case, we take the transfer. In this case, the transfer of genome is from a parent cell. So that parent cell will transfer its genome by replicating its genome and then passing it on to an offspring. And this is done by binary fission in the bacterial cell. And this is a common type of gene transmission found in prokaryotic and eukaryotic cells. Another way that genes are transmitted, though, and found in bacterial cells is HGT, horizontal gene transmission. And this is basically the transfer of genetic elements, not from parent cell to offspring, but from one cell to another cell of the same generation. So basically, a bacterial cell is sharing its genome with its neighbor, with another bacterial cell found in that same generation within that environment. And how that's done is it transfers its mobile genetic element, movable genetic element, to a neighboring bacterial cell. That recipient bacterial cell then will acquire those new genes and traits. And once it acquires those new genes and traits, it may be passed on to future generations. So, mobile genetic elements, or MGEs, definition are those genetic elements that can move around within a single cell or that may be transferred between cells of the same generation. So those, whenever you see an MGE, think of mobile genetic elements, okay, or genetic elements or pieces of genomes that may be transferred within that cell or onto or between cells. These transposable elements were first discovered by Barbara McClintock in 1931. And the types of mobile genetic elements found in bacteria are plasmids, your DNA transposons, retrotransposons, and bacteriophage elements. So we're going to be talking about 3 processes in lecture 3, part 3 of this lecture, that will go into further detail on how this can occur. A little bit more about plasmids. You know that plasmids are extrachromosomal DNA. They're found in bacteria, rarely in eukarya or archaea. So they are extrachromosomal DNA, which means that they're in addition to the chromosome. So here we've got two bacterial cells from the genus Bacillus. On the left is Bacillus cereus, and on the right is your pathogen, Bacillus anthracis. So they belong to the same genus. If you notice Bacillus cereus, it's got the chromosome, but no plasmids. The Bacillus anthracis has a chromosome, but if you notice, it's got two additional circular units, and these are plasmids. The one plasmid, PXO1, encodes for toxin, so once it's expressed, it makes a toxin, and that toxin suppresses the host immune system. The smaller purple plasmid, the PXO2, also, when expressed, encodes for a capsule. And that capsule actually will protect the bacterium from the host immune system. Once it has a capsule, the host immune system cannot attach to that bacterial cell. It basically, capsule will protect it by hiding it, by not allowing the host immune cells to attach to it. So both of these plasmids do help the bacterial cell in infection, and it makes it pathogenic. There are several other types of plasmids, and they are in your textbook, but I will mention them here. We've got F plasmid, which aids in fertility, aids in the transferring of its genome. There are R plasmids, which is the letter R, and this aids in resistance. Okay, and what kind of resistance would be like your antibiotic resistance? Virulence plasmids, they usually carry a gene for toxins, which is what this Bacillus anthracis has. These are examples of virulence plasmids. And then finally, degradation plasmids. This helps bacteria digest compounds that are not usually found in the environment, such as toluene and xylene. Actually, the type of bacteria that do carry these degradation plasmids are beneficial in industry. They use some of these bacteria to digest or to break down oil spills on oceans. We've used these types of bacteria containing these type of plasmids, so they can be helpful, the ones with the degradation plasmids. Now, transposable elements or transposons are a type of MGE, and what these mobile elements do is basically they can move within the cell, move genomes within a specific bacterial cell. And we have two types. We have the DNA transposons and the retrotransposons. So in the case of DNA transposons, a little segment, piece of genome is cut out from the DNA by an enzyme transposase, and then it can move to another area on that chromosome within that bacterial cell. Or in some cases, that DNA may be cut out by the transposase and move within that cell from the chromosome to a plasmid that may be in that cell. In the case of your retrotransposons, you look at that word retro, yes, think of a certain type of enzyme. The DNA is first transcribed into RNA, and then that RNA is then reverse transcribed by the enzyme reverse transcriptase, there's that guy, back into a complementary piece of DNA or cDNA, and then that that complementary DNA will move to another spot within that chromosome, within the bacterial cell. So again, these are two types of transposons, or a transposon is a type of mobile element in bacterial cells. The bacteriophage, we know that it is a virus, and the bacteriophage also can transfer a bit of the bacterial cell's genome to a new bacterial cell, or to another bacterial cell. And they do this by picking up genes from the bacterial cell, and they will take it along with their genome to another bacterial cell. And again, this bacteriophage will work into, or is a big part of, a type of process, of one of the three processes that I will talk about in the next lecture. So finally, that's where I'm going to stop today. Part 3, I will talk about the specific processes of gene transfer. And stay tuned, and I will see some of you later today.